Okay, so the N-terminal is the start of the protein, and the C-terminal is the end of the protein. And the convention is to write the sequence from N to the C-terminus. So let's talk about proteins. So the primary structure, what is the primary structure of a protein? It's the order of amino acids connected by peptide bonds. So why is this important? Because the sequence of these amino bonds, of these amino acids, determines the 3D structure of the proteins, and the structure of the protein gives rise to the function of the proteins. So that's why we're learning about all of this and why it's so important. Okay, so now let's talk about the actual structure of this peptide bond. Peptide bonds have a double character, right? So right here, you see that, well, let's compare it to the original, right? Right here you see that there's a double bond, right? So this is the bond, peptide bond. You have a double bond between the carbon and the oxygen and then the nitrogen, right? So all I'm saying here, right, is I'm saying that between this bond, there's a double bond character, right? Because the lone pair can form a double bond, and then this double bond can form a negative charge. So it has a double bond character through this entire N and oxygen, even though it looks like in the original one just a single bond here. So all this is saying is that through this entire bond, and right here too, is that peptide bonds have a double character. And so this is really important because that makes them planar. And so rotation around this is forbidden. So when you have a single bond, it can rotate anywhere it wants. But when you have a double bond, it rotates together. And so rotation is forbidden around these bonds. Okay, so another thing is the transform of the peptide bond is preferred because the cis causes steric clashes. So let's talk about that. So here's the bond in the transform, right? So you have the peptide bond right there. This is the transform, and over here, this is the cis form. So notice how over here it's pretty like clear and open, but here where the blue marks are, there will be steric clashes. And so obviously this isn't good because these two things will hit each other, and that's not favored in real life. It's favored when everything's as far apart as possible. And so these two models might look confusing. So if you remember back to organic chemistry, when you have a trans structure, just a simple trans structure like this is a lot more convenient when these two things are as far away as possible than when it's over here, right? If these two, oh, and another thing is if these were double bonds, right? So let me draw that because that makes more sense. So you have a double bond here and here, right? So when you have a double bond like this, these two things want to be as far away as possible. And over here, this is just not favorable because these two bulky side groups are just too close to each other and they're interfering, and nature doesn't like that. So that's why the transform is favored in general for peptide bonds. But there is one exception, and that's proline. So why is that? So for proline, if you remember, proline is the ring amino acid, and so when you have the peptide bond, you have steric clashes in the trans form over here, but you also have steric clashes in the cis form over here. So therefore, the trans form actually isn't necessarily preferred in the case of proline, and so that's the one exception to this transform preference. Okay, so let's talk about secondary structural elements. So there's three people involved in proposing the structure of the alpha helix: Linus Pauling, Robert Corey, and Herman Branson. So all three people proposed the structure of the alpha helix, and they also proposed a gamma structure, but the gamma structure actually has never been found, and probably will not be found. But this alpha helix structure was actually surprisingly correct, given the fact that they didn't have that much information to work with. So let's talk about the alpha helix. It's stabilized by the backbone hydrogen bonding. The side chains actually extend outward from the helix, and there's 3.6 amino acids per turn. Okay, so let's look at what all that means, right? So you have your polypeptide chain, right? And so what is it talking about when it's talking about hydrogen bonding? Well, each backbone NH bond, right? So you have nitrogen and hydrogen right there, is bonded to the backbone CO group of the fourth preceding amino acid. So notice how this bond, right, of the fourth, so this is the fourth amino acid, is bonded to the first one, right, the first amino acid. So it's bonded like that. So 4 amino acid, the fourth preceding amino acid. So that's when it's talking about hydrogen bonds over here, stabilized by backbone hydrogen bonding. Okay, so another thing is that alpha helices are right-handed. So what does that mean? Well, if you take your right hand out and curl it inwards, that's how it will spin, right? And so just the same way as when you're curling in your right hand, that's the same way this alpha helices is curling in, right? And so let's also talk about the other secondary element, which is beta strands or beta sheets. So they come in 3 different types. You have parallel, anti-parallel, and mixed. There's also another type of secondary structural element called the reverse turn. So why are these needed? They're needed for sheets and for helices to pack with each other. So that's a pretty important function. And how is this done? Well, it's done by the hydrogen bond from a backbone NH to the backbone CO of the third preceding amino acid. Remember that the previous one is the fourth preceding amino acid right here. So that's how the reverse turn is different, is it's linking to the third preceding amino acid. So most turns have small R groups or groups that can further stabilize the turn by bonding. This makes sense because you want that tight turn, and so for that you would need a small side chain instead of a large bulky side chain. Okay, so, and then another structural element are loops. So what do? They allow the chain to change direction. They lie on the protein surface. There's no regular periodic structure, and sometimes the loops contain reverse turns. So the next level is the tertiary structure. These are determined by X-ray crystallography and NMR spectroscopy. So this is important because if you think about it, how would you determine the 3D structure of such a tiny little thing, right? And it's using these 2 types of technologies. So once again, why is this important? Because if you know the tertiary structure, the sequence will determine the structure of the protein, and then it will lead to the function of this protein. So what are some protein structure stabilizing factors? So a few of them are disulfide bonds, hydrogen bonds, ionic bonds, van der Waals forces, and hydrophobic interactions. So let's talk more about the hydrophobic effect. So the hydrophobic effect is when you have 2 nonpolar molecules and then you have water, right? So obviously these don't interact well, so the water will form a cage around the nonpolar molecule. And so the problem with that is this is entropically disfavored. So if you remember, entropy is where it favors disorder. So this is too much order, right? Because they're all neatly organized around the nonpolar molecule. So nature doesn't like this. So what it will do is it will force these 2 nonpolar molecules to stick together. Therefore, if you notice, there's less water around the molecules entirely, right? For example, there's so many water molecules here, but when they're forced together, there's very few water molecules. And the rest of the water molecules can be a lot more disordered. And so this is favored in nature. So greater entropy is more favorable. So why is this important? Well, the hydrophobic effect leads to protein folding because it goes from the unfolded ensemble to the folded ensemble. And this is important because on the interior side is where all the hydrophobic proteins are and all the hydrophobic R groups are. So this is how the protein folds into the correct shape, and that's how you get the special structure of the protein and then therefore the function of that protein. So the final level is the quaternary structure. And so for this level, all it is the spatial arrangement of multiple subunits, and the subunits can be the same or different. So all it means is it's made up of a different bunch of different parts, and those parts can either be the same subunits or they can be different subunits joined together. So I hope that helped, and thanks for watching.